Wonderful. So my name is Jayson Jeganathan, and I'll be talking about functional alignment. So in this talk, in the first part, I'll actually introduce this topic, this exciting methodology of functional alignment, or hyperalignment. And in the second part, I'll introduce a new method to combine anatomical and functional landmarks for aligning people's brains. Now, standard anatomical alignment assumes that the same location is going to be doing the same function in different individuals. But we know that quite often this is not the case. If you look at people's brain activations for a common stimulus, the activation can be located differently in different people. Now you might say, hey Jayson, there's already a method to deal with this, and this is multimodal surface matching. And that's kind of true. So we have in the Human Connectome Project dataset alignment using MSM-all. Now, MSM-sulc, over on the left in the picture up there, is where people's brains are aligned using gyri and sulci and the overlap between them. MSM-all includes resting state fMRI to the alignment. And what this picture is showing is that the brain activations for a working memory task are sharper if you use MSM-all on the right compared to MSM-sulc on the left. However, the multimodal surface matching methodology requires that the mapping from one individual to another is diffeomorphic. And what that means is that the differences from me to Ruby, for example, have to be a warp or a twist in the brain surface. It can't be a flip or a tearing. But we now have reasonable evidence to say that this restriction might be too restrictive. So there was this method of hyperalignment or functional alignment pioneered by the lab of James Haxby and colleagues, which relaxes this assumption. And here we allowed the mapping from one individual to another to be non-diffeomorphic. Let me illustrate with a small schematic. So in the picture on the right, you see the brains of two individuals, and you see single cortical parcels with 6 voxels in them, because it's a cartoon. And both of these individuals have watched the same movie. And the colored boxes in those brains are showing which voxels lighting up for particular moments in the movie. So the yellow box, for example, corresponds to the moment in a movie where something particular happened, like they saw a dog or something. And what I want to draw your attention to is that when people saw a dog, it was the first voxel, the top voxel, that lit up in subject X, but in subject Y, it was the 5th voxel. So in a sense, you might say that these voxels are doing the same thing in the different individuals. Functional alignment essentially aims to find a transformation matrix to convey that information. And that's the matrix you see on the right, on the bottom, which is labeled R. You can see that the R matrix has a 1 in the first row and 5th column. And that's basically saying that the first voxel in subject X is doing the same thing as the 5th voxel in subject Y. Now, this matrix is a permutation matrix. It has a lot of zeros everywhere, but we don't necessarily need to be that restrictive. There are different methods, like Procrustes alignment or optimal transport, that can discover a more dense matrix, where we say that a single voxel in subject Y is predicted by a linear combination of voxels in subject X. Now, how do we know this is doing anything useful? So we can test it. For example, we've tested it in the Human Connectome Project data. On the bottom, you see the fMRI, movie viewing fMRI of 3 different individuals. On the top, you see the templates movie viewing fMRI time series. And this can simply be the group mean of a bunch of people's movie viewing time series. And we use this hyperalignment or functional alignment method to discover these R matrices that are going to map from each individual to the template. So we've got Rx, Ry, Rz. And the question is whether these R matrices are going to be doing anything useful for us. And so what we do next is that we use those R matrices to transform an out of modality, a different modality of data. In this case, we take task fMRI, and which has already been aligned with MSM-Salk, for example, and we multiply this data by the subject-specific transformation matrix. And that yields, on the bottom, these functionally aligned task fMRI maps. Now, if functional alignment is doing anything useful, we're going to see that the alignment between subjects is better after the functional alignment as compared to before. And you can measure this in a couple of ways. One of the ways would be looking at the inter-subject correlation. The other way would be looking at classification accuracy, training some sort of decoder or task classifier on the data in the bottom. So this here is a plot, again from the lab of James Haxby, showing on the top that inter-subject correlations are not that great if you have anatomical alignment alone. But on the bottom, if you use hyperalignment, the inter-subject correlations are much improved. This plot here shows my replication of those kinds of results using surface-based human connector and project data. The horizontal axis is inter-subject correlation, and each dot corresponds to a single subject. So here we're measuring whether a single subject's kind of pattern of activations for different tasks is similar to all the other subjects. At the top, in orange, you have MSM-Salk only, so that is with anatomical alignment. And on the bottom, we have functional alignment. And what you can see is that for almost every single subject, the inter-subject correlations are better after functional alignment as compared to with MSM-Salk alone. Now, you might also ask, why have I labeled the bottom, the blue group as MSM-Salk and functional alignment? Now, that's just to kind of illustrate to you that this functional alignment is something you do to data that's already been registered in a rough way with MSM-Salk. So the functional alignment is a sort of scrambling of vertices that you do afterwards. Now, on the bottom is a similar outcomes that you can see if the outcome is classification accuracy instead of inter-subject correlation. Here, each dot is a fold in cross-validation rather than being a single subject. So that ends my introduction to functional alignment. And now I'll tell you about a new method that we've developed. So we've already talked about some of the disadvantages of anatomical alignment only at the top. And that is that it ignores the individual differences in functional topography. However, there are also disadvantages to functional alignment. And crucially, one of the disadvantages is that it lets go or discards potentially useful anatomical constraints. And so we thought, why not kind of do a bit of both? And there are good reasons we might want to do that. For example, if the fMRI data is quite noisy, then ideally we wanted a method that would just say, let's ignore that fMRI data and just stick to the good anatomical alignment that we've already got. So how do we combine these two things? So on the bottom, you can see the R matrix that I talked about before. And let me remind you that functional alignment involves taking data that's already aligned, for example, with MSM-Salk, and multiplying it by this R matrix on the bottom right. But you can kind of position pure anatomical alignment within the same framework by just imagining that you take MSM-Salk data and you multiply it by an identity matrix on the top, which does nothing. There's no scrambling at all. So we thought, why not develop a method that interpolates between these two transformations to find a happy in-between? And that was quite simple to do. We simply interpolated with a simple hyperparameter, which we called gamma. So if gamma is 0 or 1, that corresponds to pure anatomical or pure functional alignment. But if you have an intermediate gamma value, that's going to correspond to this new method that we called integrated alignment. We then tested whether integrated alignment would do any better than the other two methods that we've already got. And indeed, it did. This is a busy slide, so let me break it into two sections. So up top, the first two rows are rows you've already seen. The first row is pure anatomical alignment. The second row is pure functional alignment. The third row in green is the new integrated alignment with the optimal hyperparameter. And what you can see is that the integrated alignment performed better than pure functional or pure anatomical alignment in almost every single individual. And the result was significant through a paired t-test. We optimized the hyperparameter in one cohort, and we tested it in a different cohort to prevent data leakage, essentially. So you might say, this is all well and good, but you really shouldn't be comparing to MSM-Sulc. You should be comparing to MSM-All. So we tried that out. And what you can see in orange is MSM-All only, and in blue, what happened when we stacked the new integrated alignment method on top of MSM-All. And what you can see here is that for almost every single individual, the intersubject correlation was improved using the stacked integrated alignment. And so this is the whole plot again together. And I'll draw your attention to one or 2 points here. You might notice that the improvements, while present for almost every single subject, are not very big compared to the spread between subjects' variability. For example, you can see this one subject on the left who is almost unalignable. And so this is true. The variability between subjects is quite large. But what I'll draw attention to is that the improvements you get with integrated alignment are probably similar in size to the improvement that you get going from MSM-Sulc to MSM-All. Here's the same comparisons when the outcome is classification accuracy instead of intersubject correlations. And all the comparisons that were significant in the previous plot were still present using this outcome measure. We also compared with a different set of functional alignment methodologies, Procrustes transformations, and it held across those validation tests. This plot here, in red, you're seeing the regions where the integrated alignment was superior to pure MSM-All. In blue, you're seeing the regions where the new method was inferior to MSM-All. And so most of the brain is red, happily for me. So to conclude, I hope that this presentation shows you how combining anatomical information and functional information for alignment is better than either method in isolation. And secondly, that a lot of the individual differences between people's brains, we're going to need non-diffeomorphic transformations to capture, and not simply warps like twisting. So in future work, we're hoping to validate this in other data sets and hoping to test the impact of these methods on brain behavior associations. If you have any more questions about this, you can come and find me at my poster on Wednesday, or you can find me on Twitter X. And I'd like to thank the whole team that was involved with this effort, particularly my PhD advisor, Michael, and all the funders who were involved. Thank you.